Hello, everyone, and welcome back to our Receptors series. In Module A1, we discussed the introduction to G-protein-coupled receptors, their structure, and their activation mechanism. We learned how ligand binding induces a conformational change in the receptor, and how this activated receptor promotes the exchange of GDP for GTP on a heterotrimeric G protein. If you have not watched Module A1 yet, I strongly recommend watching it first, as the concepts covered there form the foundation for everything we will discuss in this module. Now that we understand how GPCRs become activated, an important question arises. If all GPCRs use essentially the same activation mechanism, how can they produce completely different cellular responses? How can one receptor increase heart rate, another reduce neurotransmitter release, and another stimulate smooth muscle contraction? The answer lies in the type of G protein that the receptor activates and the intracellular signaling pathways that follow. In this lesson, we will answer three key questions. How do GS, GI, and GQ proteins produce completely different cellular responses? How do GPCR signaling pathways work after receptor activation? And what roles do second messengers such as cyclic AMP, Inositol triphosphate, Diacylglycerol, DAG, and calcium play in transmitting and amplifying cellular signals? Before discussing GPCR signaling pathways, it is important to understand how GPCRs span the plasma membrane 7 times. One of the most distinctive features of G-protein-coupled receptors, or GPCRs, is that they span the plasma membrane 7 times. But how is this possible? The answer lies in the receptor's protein structure. A GPCR is made up of a single polypeptide chain that contains 7 hydrophobic regions. These regions are rich in nonpolar amino acids, making them compatible with the hydrophobic interior of the lipid bilayer. As the receptor is synthesized and inserted into the cell membrane, each hydrophobic region folds into an alpha helix and becomes embedded within the membrane. Each alpha helix crosses the membrane once. Since there are 7 of these transmembrane helices, the receptor passes back and forth through the plasma membrane 7 times. This creates the characteristic serpentine, or snake-like structure that is typical of all GPCRs. As a result of this arrangement, the N-terminus of the receptor is located on the extracellular side of the membrane, while the C-terminus is located inside the cell. The 7 transmembrane helices are connected by 3 extracellular loops and 3 intracellular loops. This 7-pass membrane architecture is essential for receptor function. When a ligand binds to the extracellular portion of the receptor, it causes the transmembrane helices to shift their positions. This conformational change is transmitted through the membrane to the intracellular side of the receptor. The activated receptor can then interact with a G protein, triggering intracellular signaling pathways and producing a cellular response. In Module A1, we have already covered the structure of GPCRs, including their extracellular, transmembrane, and intracellular domains, and how these structural features enable signal transmission across the plasma membrane. Let us now examine how GS, GI, and GQ proteins produce different cellular responses. Although all GPCRs share the same basic activation mechanism, the cellular response depends entirely on the identity of the activated G-alpha subunit. Once activated, the G-alpha GTP complex dissociates and interacts with specific intracellular effector enzymes. This is where signaling pathways begin to diverge. Different G-alpha proteins regulate different effector enzymes. These effector enzymes generate different second messengers, activate different protein kinases, and ultimately trigger distinct cellular responses. Therefore, while the initial receptor activation process is similar for all GPCRs, the downstream signaling cascade depends on whether the receptor couples to GS, GI, or GQ. The 3 major GPCR signaling pathways are: the GS pathway is generally stimulatory, the GI pathway is generally inhibitory, the GQ pathway, which activates phospholipase C and generates a completely different set of second messengers. Let us begin with the GS pathway. GS stands for G-stimulatory protein. As its name suggests, its primary function is to stimulate the activity of the enzyme adenylyl cyclase. Adenylyl cyclase is embedded within the plasma membrane and serves as the major effector enzyme of the GS pathway. Next is the mechanism of GS signaling. The signaling process begins when a ligand binds to a GS-coupled receptor. Examples include the beta-1 adrenergic receptor, beta-2 adrenergic receptor, and glucagon receptor. Once activated, the receptor stimulates the GS protein. The G-alpha-S subunit exchanges GDP for GTP and becomes activated. The activated G-alpha-S GTP complex then binds directly to adenylyl cyclase and stimulates its enzymatic activity. Activated adenylyl cyclase rapidly converts ATP into cyclic AMP. This marks the beginning of a powerful intracellular signaling cascade. As intracellular cyclic AMP levels rise, cyclic AMP binds to and activates protein kinase A, or pKa. Activated pKa then phosphorylates numerous target proteins throughout the cell, including metabolic enzymes, ion channels, structural proteins, and transcription factors. Through these phosphorylation events, pKa alters cellular metabolism, modifies ion channel activity, regulates gene expression, and changes overall cellular behavior. As a result, activation of the GS pathway typically produces stimulatory effects such as increased heart rate, increased cardiac contractility, enhanced glycogen breakdown, stimulation of lipolysis, increased hormone secretion, and enhanced gene transcription. Overall, GS signaling promotes cellular activation. Next, we have the GI signaling pathway. While GS activates adenylyl cyclase, the GI pathway does exactly the opposite. GI stands for G-inhibitory protein. Like Gs, it targets adenylyl cyclase, but instead of stimulating the enzyme, inhibits its activity. Now, let's see the mechanism of GI signaling. The pathway begins when a ligand binds to a GI-coupled receptor. Examples include the alpha-2 adrenergic receptor, M2 muscarinic receptor, and dopamine D2 receptor. Following receptor activation, the GI protein is activated by GDP-GTP exchange on the G-alpha-I subunit. The activated G-alpha-I GTP complex then binds directly to adenylyl cyclase and suppresses its enzymatic activity. As adenylyl cyclase activity decreases, cyclic AMP production falls. Lower intracellular cyclic AMP levels mean that protein kinase A remains inactive or significantly less active. Consequently, downstream phosphorylation cascades are suppressed, and overall cellular activity is reduced. Additional actions of the G-beta gamma subunits. An important feature of many GI-coupled receptors is that signaling is not limited to the G-alpha subunit. The released G-beta gamma subunits can also participate in signaling. In many cardiac and neuronal cells, these G-beta gamma complexes activate G protein-coupled inwardly rectifying potassium channels, often called G protein-coupled inwardly rectifying potassium or GIRK channels. Opening these channels allows potassium ions to leave the cell, making the membrane potential more negative. This process, known as hyperpolarization, decreases cellular excitability and makes it more difficult for the cell to generate an action potential. Cellular effects of GI. As a result, GI signaling typically produces inhibitory physiological effects, such as slowing of heart rate, reduced neurotransmitter release, decreased hormone secretion, reduced metabolic activity, and overall suppression of cellular function. Now that we have examined both the GS and GI pathways individually, let's briefly compare them. Although both pathways act on the same effector enzyme, adenylyl cyclase, they regulate it in opposite ways. GS stimulates adenylyl cyclase, leading to increased cyclic AMP levels and activation of pKa. In contrast, GI inhibits adenylyl cyclase, resulting in decreased cyclic AMP levels and reduced pKa activity. As a consequence, GS generally produces stimulatory cellular effects, whereas GI produces inhibitory cellular effects. So, despite using the same signaling components, these pathways generate opposite physiological responses. This comparison highlights how the specific G protein coupled to a receptor determines the final cellular outcome. The third major pathway, GQ, introduces an entirely different signaling strategy. Unlike GS and GI, GQ does not primarily regulate adenylyl cyclase or cyclic AMP production. Instead, it activates a different effector enzyme, phospholipase C-beta. Next is the mechanism of GQ signaling. The pathway begins when a ligand binds to a G protein-coupled receptor. Examples include the alpha-1 adrenergic receptor, M1 muscarinic receptor, M3 muscarinic receptor, and histamine H1 receptor. Following receptor activation, the GQ protein becomes activated through GDP-GTP exchange on the G-alpha-Q subunit. Activated G-alpha-Q GTP then binds to phospholipase C-beta and stimulates its activity. PLC-beta acts on the membrane phospholipid, phosphatidyl inositol 4-5-bisphosphate, or PIP2. Although PIP2 represents only a small fraction of membrane phospholipids, it plays an extremely important role in cell signaling. When activated, PLC-beta hydrolyses PIP2 into two second messengers, Inositol 1,4,5-trisphosphate, or IP3, and Diacylglycerol, or DAG. These two molecules initiate separate but coordinated signaling pathways that ultimately converge to activate Protein Kinase C. The GQ pathway ultimately produces its effects through the combined actions of IP3, diacylglycerol, calcium ions, and Protein Kinase C. Unlike the GS and GI pathways, which primarily regulate cellular activity through changes in cyclic AMP levels, the GQ pathway controls cellular function through calcium signaling and PKC activation. As IP3 stimulates calcium release from intracellular stores and diacylglycerol activates protein kinase C, numerous cellular processes are affected. The cellular and physiological effects of GQ signaling include smooth muscle contraction, glandular secretion, neurotransmitter release, activation of metabolic enzymes, regulation of gene expression, cell growth and proliferation, inflammatory responses. Because calcium serves as a universal intracellular signaling molecule, activation of the GQ pathway can influence a wide range of cellular functions in many different tissues. For example, activation of alpha-1 adrenergic receptors in vascular smooth muscle causes vasoconstriction, while activation of M3 muscarinic receptors in exocrine glands stimulates secretion. Overall, the GQ pathway is particularly important for controlling contraction, secretion, metabolism, and long-term cellular responses. Now, let us shift our focus from the G protein subunits themselves to the real signal carriers inside the cell. We will now explore the major second messengers, cyclic AMP, IP3, diacylglycerol, and calcium, and understand how each one translates receptor activation into specific cellular actions. When a signaling molecule binds to a receptor on the cell surface, it acts as the first messenger because it remains outside the cell. Its role is to initiate communication at the plasma membrane. However, this information must be relayed to intracellular targets located throughout the cell. This is achieved through a group of intracellular signaling molecules known as second messengers. Second messengers are small, rapidly acting intracellular molecules generated following receptor activation. They transmit and distribute signals from the plasma membrane to intracellular effector systems. A key feature of second messengers is their ability to convert a localized membrane event into a widespread intracellular response. Because they are small and often diffusible, they can rapidly spread the signal within the cytoplasm. The major second messengers involved in GPCR signaling are cyclic AMP, inositol 1,4,5, trisphosphate, diacylglycerol, and calcium. Together, these molecules function as intracellular signal mediators that determine how the cell interprets and executes receptor-generated information. Second messengers are essential because they define the intensity, distribution, and specificity of intracellular signaling. They amplify signals generated at the cell surface. They ensure rapid spread of information within the cytoplasm. They activate intercellular regulatory proteins and enzymes. They control downstream cellular responses, such as metabolism, secretion, and gene expression. They coordinate multiple cellular processes simultaneously in response to a single extracellular signal. Without second messengers, receptor activation would remain restricted to the membrane and would not produce effective intracellular responses. Instead, second messengers allow a single signaling event to be translated into a coordinated cellular response. Let us begin with one of the most important second messengers in biology, cyclic AMP. Cyclic AMP is one of the most important intracellular second messengers in GPCR signaling. It functions as a rapidly diffusible molecule that converts extracellular signals into intracellular responses. Cyclic AMP is synthesized from ATP by the enzyme adenylyl cyclase. Its intracellular levels are tightly regulated, allowing dynamic control of cellular signaling strength. The major target of cyclic AMP is protein kinase A. Cyclic AMP binds to the regulatory subunits of pKa, releasing the catalytic subunits and activating the enzyme. Activated pKa phosphorylates a wide range of intracellular proteins involved in cellular regulation. Through pKa activation, cyclic AMP regulates metabolic pathways, ion channel activity, gene transcription, hormonal responses, signal integration and amplification. Cyclic AMP therefore acts as a central coordinator of intracellular responses to extracellular signals. Cyclic AMP signaling produces strong amplification effects. Small changes in receptor activity generate large amounts of cyclic AMP, which activates multiple pKa molecules, each acting on many downstream targets. This allows weak extracellular signals to produce strong cellular responses. The next important second messenger is inositol 1,4,5 trisphosphate, commonly called IP3. Is a key second messenger involved in calcium mobilization within the cell. It links membrane-derived signals to intracellular calcium release. IP3 is generated from the membrane phospholipid, phosphatidyl inositol 4,5-bisphosphate, PIP2, and rapidly diffuses through the cytoplasm. IP3 binds to IP3-gated calcium channels on the endoplasmic reticulum, triggering the release of stored calcium into the cytoplasm. IP3 increases intracellular calcium concentration, which regulates muscle contraction, secretion processes, enzyme activation, and cellular signaling pathways. IP3 functions primarily as a calcium-releasing messenger. IP3 does not directly cause cellular effects. Its role is to release calcium, which acts as the main downstream signaling molecule. The second messenger generated alongside IP3 is diacylglycerol. Unlike IP3, DAG does not diffuse through the cytoplasm, as it is a lipid-based second messenger that remains embedded in the plasma membrane. It all begins with the membrane phospholipid, PIP2. When phospholipase C is activated, it cleaves PIP2 into two second messengers, IP3 and DAG. While AP3 diffuses through the cytoplasm, DAG remains embedded within the plasma membrane. Meanwhile, PKC exists in an inactive form within the cytoplasm. Following IP3-mediated calcium release from the endoplasmic reticulum, calcium helps recruit conventional PKC isoforms to the plasma membrane. PKC contains two important regulatory domains, a C1 domain that binds DAG and a C2 domain that binds calcium. Once PKC reaches the membrane, DAG binds to this C1 domain, anchoring PKC at the membrane and inducing conformational changes that promote its activation. Activated PKC then phosphorylates specific target proteins, triggering signaling pathways involved in cell growth, differentiation, secretion, metabolism, and gene expression. Thus, the two products of PIP2 cleavage work together. IP3 increases intracellular calcium levels, while DAG binds the C1 domain of PKC, helping localize and activate the kinase at the plasma membrane. Among all second messengers, calcium is one of the most versatile and universally used signaling molecules in biology. Under resting conditions, intracellular calcium concentrations remain extremely low. This allows even small increases in calcium levels to function as powerful signaling events. Once released into the cytoplasm, calcium produces its effects by binding to a variety of regulatory proteins. These proteins act as intracellular sensors that detect changes in calcium concentration and convert them into specific cellular responses. One of the most important calcium-binding proteins is calmodulin. When calcium binds to calmodulin, the protein changes shape and becomes activated. Activated calmodulin can then regulate numerous enzymes and signaling pathways involved in metabolism, secretion, cell growth, and gene expression. Calcium also activates various protein kinases, particularly calcium calmodulin-dependent protein kinases, or CAMKs. These kinases phosphorylate target proteins, allowing calcium signals to influence a wide range of cellular activities. In muscle cells, calcium binds to contractile proteins such as troponin. This interaction initiates the molecular events that lead to muscle contraction. Finally, many calcium-dependent enzymes require calcium binding to become fully active. Through the regulation of these enzymes, calcium can directly control important cellular processes, including metabolism, secretion, and intracellular signaling. Calcium regulates muscle contraction, neurotransmitter release, hormone secretion, enzyme activity, and gene transcription. Calcium acts as a universal intracellular regulator. At the beginning of this lesson, we asked an important question. What roles do second messengers such as cyclic AMP, IP3, DAG, and calcium play? The answer is that second messengers function as intracellular signal carriers and signal amplifiers. Cyclic AMP activates protein kinase A. IP3 releases calcium from intracellular stores. DAG activates protein kinase C, and calcium regulates contraction, secretion, metabolism, enzyme activity, and gene expression. Together, these molecules transform an extracellular signal into a coordinated intracellular response. This brings us to the end of Module A2 on specific G protein pathways and second messengers. In the next lesson, Module A3, we will move from basic mechanisms to clinical and pharmacological applications. We will compare GPCRs with ligand-gated ion channels, examine major clinically important GPCR examples, explore their roles in human disease, discuss their pharmacological relevance, and conclude with high-yield revision points, especially useful for examinations and clinical practice. If you found this lesson helpful, make sure to like, share, and subscribe for more lectures. Thank you, and see you in the next video.